Kiran and Hriday, please allow Renuka and Prash. Yeah. Hello.
Sir, we have not received the request.
He says that. Okay, I will ask him to use the link you have given me.
It's the same link which Kiran has shared with everyone.
Okay. Kiran?
I haven't received the request, so they have to give the email IDs. So I just written. So whoever is not getting a request, they should give the email IDs. Renuka is Bhaveshri and they are ready. I made you as host, so you should be able to talk.
Yes. Hi, all everyone. Hi, Kiran. Yes, both are ready. And
Okay.
can I switch on the video, right?
Sure, you can.
Uh, so we are starting at 8:30 or
Yes, 8:30.
okay.
I'll announce that.
Yeah. So I will
Sure.
just start at 8:30.
Yeah, just be ready. Will it be only one ID or also another with the Prash ID also you will speak or you will do this?
Yeah, with the same ID.
Ya kun.
Yes, yes, Renuka, please go on. So thank you, everyone. So we start with Saraswati Vandana, Bhavyashree, and Nirmala.
Ya kundendu tushar dhavana, ya shubhra vastravrita, ya veena varadanda mandakara, ya shveta padmasana, ya brahmachatram प्रभृति देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजात्यापः शुक्लां ब्रह्मविचारसार परमा आद्यां जगद्वाणी वीणा पुस्तकदान ऐं अध्यादां ज्यादादकदां हस्ते स्फाटिकां विधाति तीं पद्मासने समस्थिताम् वन्दे तां परमेश्वरी भगवती बुद्धिप्रदां शान्तराम्. Thank you. Thank you.
Thank you, Bhavyashree, and thank you, thank you, Nirmala. So let's go ahead with a nice start with a Saraswati Vandana. And thank you, everyone, for joining to this Galaxy pre-conference workshop in BIX 2021. Now, Jairaman sir will be introducing about the chair, and he'll be the chair and introduce about the Narahari Shastri sir with the opening remarks.
Good morning, everyone. On behalf of INBIX 2021 and Galaxy Organizing Committee, I invite you all warmly for the 2-day exciting sessions on the pre-conference Galaxy workshop. The opening remarks would be given by Professor Narahari Shastri. I will say a few words about him before he gives the remarks. Dr. Shastri is currently heading the CSIR Northeast Institute of Science and Technology, Jorhat. Originally a chemist, he has made fundamental contributions in the areas of computational and theoretical chemistry, theoretical organic chemistry, and reaction mechanism. Also on software and database development for drug discovery, and on different aspects of non-covalent interactions. And his research work was published in about 300 research papers and reviews, which received over 11,000 citations. Among many awards, Professor Shastri was bestowed upon Shanti Swarup Bhatnagar Prize in Chemical Sciences in the year 2011 is the most coveted. Other noteworthy awards include Swarna Jayanti Fellowship in 2005 by DST, B.M. Birla Award in the year 2001, B.C. Deb Memorial Award in the year 2009, CRSI Medal in the year 2011, and Alexander von Humboldt Fellowship. He is also a Sir J.C. Bose National Fellow. He is a Fellow of National Academy of Sciences, Fellow of the Indian Academy of Sciences, Fellow of Royal Society of Chemistry, Fellow of the Association of Biotechnology and Pharmacy, Telangana State Academy of Sciences, and Andhra Pradesh Academy of Sciences. He is also a Fellow of INSA, and thus he has a rare distinction of becoming a Fellow of the 3 most prominent science academies of India. He also was recently elected as a Fellow of the Biotech Research Society, India. It is very apt that Professor Shastri, with a strong background and experience in computational chemistry, provides the opening remarks in Galactic Workshop and declares the workshop open. Professor Shastri, sir.
Thank you, sir, Professor Jayaraman, and good morning, all of you. After we kickstart the meeting with nice rendering by Bhavya and Nirmala, we have to celebrate knowledge. And I would like to see that knowledge or science is done not because and not necessarily because it is useful, because it can be done. It is very important to be able to do science and create a new space which was not existing before. When we started this organization called Bioclues, it all has started with some enthusiasm by a group of people, primarily led by Prash and a few others, and it sustained almost 12 to 14 years. And a little bit before that, in 2005, Galaxy also has started, where it became open in 2007. And the idea of Galaxy is also to make the science open. Although it started from omics research, the tool Galaxy is much more profound than some of the areas for which it is developed. And I am very happy to see that the talks that were lined up, the keynote by Gareth Price on introducing the Galaxy community and the ecosystem, followed by the work on the NGS, which is the main focus of the Galaxy, and also epigenetics, metagenomics, transcriptomics, and omics in general, where the platform is very, very strong. And I'm very happy to see that the structural bioinformatics and chemoinformatics also, while not so much used in Galaxy by the community who developed, this is very powerful too. This is followed by a talk on protein ligand docking and chemoinformatics. And afterwards, there's a lecture from my group where we have been using the Galaxy for drug discovery software, the molecular property diagnostic suite. And there are other talks on proteogenomic workflows using Galaxy and epigenome analysis. I strongly believe that this platform is a very powerful platform where people can write different scripts, and then the workflow system and the openness of the entire community and the philosophy will make it very, very useful for the science in general and computations in biology in particular. And I feel that there can be multifaceted utilities that we can take from this Galaxy platform. With this, I would like to declare the conference open, and I would like to say a few words about Bioclues. And I am very happy that this particular organization is withstanding and going merrily in the last 14 years, although the support system is not very much there. The support system of this particular organization is just the people and their investment. And I'm sure that we will have to really see that how this impacts bioinformatics in the world in general and in our country, India in particular. And one of the primary, I was trained as a computational chemist using high-level quantum chemical calculations on small molecules. And when I started my PhD, molecules of the size of 10 atoms is too big, which were very difficult for them to be subjected to rigorous computational modeling like ab initio and post-artifact calculations. But now the computational modeling on large systems with different algorithms are coming up in a very big way. And biology is definitely requiring computational modeling and in its more rigorous form. But when we have the science itself as driven by the paradigms of experiment, because doing experiment is important. Only thing, according to a statement, that everything is theoretically impossible unless it is practically done. So you can develop theories or you can do experiments. Then these are two different and fundamental paradigms of science. And when computers and the theories have developed, which has metamorphosed into algorithms and software programs, modeling has become a stabilizing third leg. And modeling has become very, very important. And that has emerged as a third paradigm of science. But the fourth paradigm of science is data science. Data science has completely changed the way in which we look at things. There is a reason for a thing to happen. This is something which is based on theory or modeling. Modeling will try to use some mathematical equations and then develop an algorithm, and there's an input and you will get an output. And for getting an output, you have an input and a set of mathematical equations. For example, if you have an input system, its position and momentum coordinates are known. After a time period t, you will be able to get the final position by solving some Newtonian mechanics. On the other hand, you know the input and you know the output, but you don't know what is the relationship between the input and an output. For example, the epidemiological studies of the emergence of COVID, or maybe the biological activity, or maybe some other aspect where there's a lot of data and there is little information or knowledge. In that way, the data science approaches have come. When you know the input and output, there are a number of ways in which we can develop a theory based on data and then come up with solutions. So this particular thing is emerged as the fourth paradigm. And in that, omics is very important. The next generation sequence is very important. But what is also very important is the chemical data. The chemoinformatics is becoming higher, and biological data, biological diversity is very, very high, and complexity is also very high. But in terms of the diversity and complexity, chemistry and biology are competing, while conventionally people say that chemistry is more diverse and biology is more complex. But the boundaries between chemistry and biology are slowly vanishing. But I just wanted to bring to the notice of younger people that when we started doing chemistry, everything is free and open source software. And most of the people who develop the software used to send us their programs where we can compile them and then use it in different machines, because there definitely you will have to compile. It's not possible to have an executable module, and there are different machines, and you have a job control language, and then you will have to spend a lot of time and make significant modifications to your source code. And if a person who develops a particular program, and if he finds that somebody else has used it, that's an achievement. But over the years, the computational modeling and computer-aided drug design or material science has become so expensive, where people have increased the prices of this commercial software to such levels that it is practically out of the reach of the many academic organizations. In contrast, bioinformatics has started with a bigger philosophy like EMBL and many other people coming up and then putting a lot of money, where most of the softwares are available for free. And I think for academic institutes, especially in colleges and in a big country like India, we need to have a little bit more orientation towards having software programs which are developed in an open source form. That has two objectives. The first objective is it will be available for free, but that's not a very big thing. We can pay some money. But most importantly, when it is open, you will have a better access to change these programs. Or if you have an idea of what is the method, you can try to make some modifications to the method. On the other hand, if you have a black box program, you may not even have to pay any attention to the theory behind this, or the methodology behind it, or algorithm behind it. So I would like all the participants to make the best use of this particular conference, and the experts are there and they will be helpful to you. These conferences will be very helpful in disseminating the knowledge, but it is also helpful to make contacts. And I strongly believe that contacts with people who are talented is very important. And if you are at a young stage, and if you are able to have contacts with people who are talented but also enthusiastic, that will help you in a long way. And if this particular conference helps at least a few of you to be really motivated to take up bioinformatics in a big way, and then try to fully utilize the strength of Galaxy. I feel that when I look at Galaxy, it is a much, much more powerful tool, and the whole use of this is a lot more than what we can think about. So that's the reason why I would like to focus this particular year's INBIX in the strengthening of open source computational drug discovery. And in that aspect, I feel that Galaxy will play an important role. But those people who are associated with Galaxy, they say, oh, it's a NGS or omics type of a thing. How in drug discovery it will be useful? Maybe by the end of this two-day workshop, and definitely during the INBIX conference, we may probably be able to get convinced that this Galaxy is not only useful for NGS and omics, but also will be of great help for computational drug discovery. I just wanted to tell you that when some techniques start, they start for a completely different reason. For example, if you take NMR has started just to look at the small molecules. And then people have looked at different isotopes. And then people have looked into 2D NMR. And there are different forms of 2D NMRs. And this has a profound influence, started from physicists, very useful for chemists, went into biology, but its big use have come in medicine. Medicine, MRI, is one of the very, very powerful techniques for NMR. And it is also very useful in defense techniques. There are so many defense applications which were being used with NMR. And similarly, if you look at the advent of quantum mechanics, people thought that, what is the use of quantum mechanics? But somebody says that currently 20% of world GDP, or even more than that, is depending on the technologies that are resultant of the quantum mechanics. Then we can go on and on. But what is very important is informatics and modeling is very important in biology. Bioinformatics is extremely important. Whatever may be the beliefs of people, without bioinformatics, we cannot do biology. I'm not saying that you have to do only bioinformatics. Even if you are an experimentalist, or if you are a biologist or a microbiologist, whatever that is, you need to have a good exposure with bioinformatics. And We have to look at the importance of interdisciplinary research or cross-disciplinary research, and most importantly, we should also think about barrierless research. We should not be in our own silos thinking that I'm a biologist, I am a chemist, I'm a physicist, I'm a mathematician, I'm an engineer, I'm a doctor, I'm an agricultural scientist. These are all superfluous barriers. And it's extremely important that we have to overcome that and then celebrate science in its fullest. And we never know what is useful in what form. Our quest is to move on and generate new knowledge and then get people who are really able to make a difference to the science at large. And I'm sure that in the next few years, some of the people who join this Galaxy workshop and INVIX conference will get full benefit out of that. And I thank all the people, especially the organizers, Kiran and Prash and the speakers. I could see that they are really able to take their time and give a lot of support. And something like this will have a lot of planning. And I also thank them for giving an opportunity for me to speak and also giving an opportunity for CSIR NEAST to organize this. I hope that everything will go well, and what determines whether it will go well or not is the active participation of the participants. Thank you very much, and I hope that the next 2 days will be extremely joyful, scientifically exciting, and people will be really enjoying these 2 days. Thank you very much.
Thank you, Professor Sastry. Galaxy is not only a platform, it's a community. Since its inception, more than 125 galaxies are available worldwide, with the big three as Galaxy Main, Galaxy Europe, and Galaxy Australia. While about the Galaxy, the respected speakers will be providing you the deliberations, I wanted to take a little time out to talk about the Galaxy India. It's a recently launched Galaxy regional community. Although the idea of launching a dedicated Galaxy India community has been thought of for more than about 10 years, it recently got an official soft launch during a Bird of Feather event during the Galaxy Community Conference, GCC 2021 in July 2021. In India, the Galaxy India community has been working in collaboration with the Bioclues Society, TMS Foundation, and CSIR to organize various training, awareness, and outreach programs from time to time. This INVIX 2021 pre-conference workshop is also one of such initiatives. The path of reaching up to the soft launch was impossible without the contribution from various people, including Professor David Clements, Simon Gladman, Gareth Price, Sonika Tyagi, Anshu Bharadwaj, Prashant Suravarjala, Pratik Jagtap, Kiran Telekunta, Kirtan Dave, Harpreet Singh, Glory Basumata, Sangram Keshari Sahu. Very special thanks indeed to Beatrice Serrano, Bjorn Gruning, and Freiburg Galaxy team for providing us an Indian instance of the Galaxy Europe server and maintaining it. We are looking forward toward the hard launch of the Galaxy India server and expanding the Galaxy community at a much faster pace with support from various organizations and with support from NEAST with the able stewardship of Professor Sastry and CSAR and various other organizations. Thanks a lot. Yeah.
Thank you, Professor Jayaraman. Yes. So I will quickly take into some kind of housekeeping things, and then subsequently the first talk will start. It might be a bit 5 minutes delay for the first talk, but I hope I will try to make it simple. And also we have a kind of pause in between, or let's say break after the first talk, then I could give much more things. So I just kept only one link here, if you see. The idea is right now to have this link to be able to do all the things. Just a minute. Okay. Yes, just. Yeah. So please have this link. I'm also putting in the chat right now in the Google Meet chat. And this link should give you more or less almost all the information. And this information will be getting updated as and when whatever the things required for any of this session. So it will be kind of a live document. You can use here. Most of the things you will get from here. So for the first thing is, I would request all of you to be in part of this Gitter channel, which was explained in the basic Galaxy Basic session also. This will help you to ask any questions. So if you are part of this, you can ask questions there. Although you can also ask questions in the Google Meet. That's also completely fine. But if you have, let's say, a kind of related to errors and other things while you're using the Galaxy, then you can use the Gitter thing. So we will have a watch on both the things, and you can use any of them. Coming back to the Galaxy, so we have, thanks to Use Galaxy Europe, they have provided a lot of infrastructure and a lot of support to us, and because of which, let's say we have a kind of exclusive let's say space. For that to work out, you should have an account in usegalaxy.eu, or if you already know that usegalaxy.in is also based on the usgalaxy.eu server. So if you go to the usegalaxy.in also, that's fine. You will land in the same. So if you have an account on usegalaxy.in or usegalaxy.eu, or it's called as usegalaxy.in is called as india.usegalaxy.eu. So all the three are same. So if you happen to have an account in any of these three instances, or let's say these three pages, not three instances, because they are all belong to same instance. And after that, you can just go and click on this link, the link which is here provided. This should be already open. It's already working. And so once you're logged in and you click on this link, then you will be having you'll be in the zone of exclusive for this workshop for the INBIX 2021. So your jobs, whatever you execute in the Galaxy, those will be taken care in a special way. So please use them. If you have any questions, do express in the question and answers or in the Use Galaxy. So then I go to the next session. So it's a great honor for me to introduce the next speaker, who is a very well-known speaker in Australian community and of course in the Galaxy community, Gareth Price. Thank you, Gareth Price, being here. And basically, I need to thank him a double thanks, because this talk was supposed to be held by Dave Clements, who is in the main center, or let's say first Galaxy used instance place. But unfortunately, he's not making it to the, say, INBIX 2021 or the Galaxy workshop. And so Gareth Price offered to replace him and give the session. And it's very, we really thank you for the double thanks on that. And so he will be introducing the Galaxy community and ecosystem. Before that, I would like to introduce him about few things. He is mainly a coordinator for the INBIX, Galaxy Australian community. He has been a genomics scientist for over 15 years. He has been involved in experimental design, assay performance, and data QC, data analysis related works. He's also in printer microarrays, cartridge-based gene chips, and multiple next-generation platforms. So he's also involved in variety of model organisms and microorganisms. And he's more recently into Galaxy and how to make data much more easily processable, let's say, data interpretation to make for the result in the discovery. So make it ready for the discovery. This is more he's in focus, and so he's very much involved in the Galaxy. But more on the Galaxy, I would not like to say. Probably he will explain more. That will make more better. So I hand it over to him. Gareth, please go on.
Thank you, Kieran. And I will share my slides as I get
Yes,
used to
please.
Google Meet, and we'll get started.
Yes.
Sorry, yep. It's just waiting for that to buffer. There we go.
Okay, no problem.
Sorry, I will do the classic online thing. You can see my slides and hear me okay?
Sorry, where are the slides?
You can or can't see them, sorry.
You could, if you want, you can give it to me and you can share it with me. That's fine.
Oh,
As
no,
an
that's
email.
all right. Sorry, it's just taking its time.
Okay, no problem.
There we go. You should be able to see them now.
Yes,
Is
it's
that-
coming up.
Yeah.
Yeah, it's coming up.
Excellent.
Great, we can go. Yeah, it's there.
Okay, thank you very much. And thank you for the kind introduction. So I have taken this talk on behalf of Galaxy Project, which I'm only slightly humbly qualified to speak on behalf of the many hundreds of fantastic people we have involved in Galaxy. My representation in this case is Service Manager of the Australian Galaxy instance, Galaxy Australia. And so there'll be a slight Galaxy Australia bias, but really I'm here to talk about the overall Galaxy community and the ecosystem that we work in. So stepping way back, what is Galaxy? Well, Galaxy Project's long-standing definition of itself is Galaxy is an open, web-based platform for accessible, reproducible, and transparent biomedical analysis. And by the Galaxy Project, I mean galaxyproject.org, shown down the bottom there. That's just one of multiple definitions that people have thrown about for Galaxy. And I think it's worth noting something like Keith Bradnam's definition. And Keith is a well-published bioinformatician who now interacts in a science communication role in London. And he's published this blog site down the bottom, 13 questions you may have about Galaxy. And I guess his definition, a web-based platform that provides a simplified interface to many popular bioinformatics tools, which is a nice, succinct, easy way to put it. But to us, and for those of you that were able to reach IMCOB yesterday, we presented some of these slides, but not all of them there yesterday. So Galaxy really is a community. And this community, to me, when I came on board into Galaxy around about 4 years ago, was something at first, I guess, I didn't really appreciate, that the strength, diversity, skill set in this community and really what it's managed to achieve. I'm very much on board now. So Galaxy is a global community, and it's a community of coders. It needs to be, because it's software. It's a community of tool wrappers that make all the bioinformatics tools and the ever-changing list of bioinformatics tools available to users of Galaxy. It's the training content developers and the trainers, and it's really a lot more. But I think it's always worth not losing sight of the fact that Galaxy is really for the scientists that use the service. Without them, there wouldn't be any point in running these fantastic services. So it's all about what science do we enable. And I'm going to finish my presentation today with some slightly indulgent but examples of science. Just bear with me, apologies. I'm just going to flip my Google Meet to another screen so I can keep an eye on everything. Right, so skipping through what it means for the code community and other communities. So Galaxy has tri-yearly updates in January, May, and September. And the September update dropped about a week ago. And again, closer to the end of the talk, I'm going to highlight some of the features there. The code community is a team of code developers, code reviewers, and committers, and they maintain the rigor around the quality of the product. And if you're interested in that code product, it's found at github.com/galaxyproject. And as galaxies grow, and I think one of the recognitions is that not everyone can do everything. So the Galaxy Working Groups is a newer part of the Galaxy ecosystem. I've shown the 12 groups down the bottom here. They range from real infrastructure and code working groups through to those training working groups, and also through to the end use, the scientific applications. So if you're interested in an element of Galaxy or understanding how it works, or have some thoughts on improvements or changes, please do engage with the working groups. It's also a tool community, and this is a really important part of Galaxy. So shown in the background is the tool shed. What you can't really see up the top is there's 8,440 tools available on October 19th. And of course, there are more than that already now. And we've seen over the years from 2015 through to 2021, a huge and consistent trending growth in the amount of bioinformatics tools available to install on a Galaxy and to enable science. Which is really covered off in these first few points. They're all hosted in a toolshed/app store, so they're navigatable and discoverable. And I think one of the important parts, and this is something that we've understood in the bioinformatics community, is a necessary part of the community, but in a positive way, is the commitment by a subsection of our tool developers. And this is the Intergalactic Utilities Commission, who are dedicated to putting rigor around the tools. And that rigor includes testing framework, documentation, training material, deployability, stability, maintenance of the tools, so that if your tool gets badged as IUC compliant, then it's a robust tool that can easily be put on any Galaxy. And I think that gives trust in the system, and that's one of the important things we're trying to build. To the point that I really wanted to emphasize this over 2 slides. We have our Hall of Fame. This is from our Galaxy training, but it's the same, essentially, Hall of Fame for our committers and our tool developers. And it's worth noting that really this is a hugely open source community, one of the largest in the world, with hundreds of people contributing constantly to the upkeep, maintenance, and development of the code base, the tools, and the training. So speaking of training, this is part of Galaxy that I interact with a great deal in my role at QCIF. So this is developing and delivering training to researchers to attempt to constantly upskill people in how to get the most out of Galaxy and how to get the most out of their data. So training at galaxyproject.org is the website. It's a freely available resource that links to all the usegalaxy.star servers globally. They all synchronize with the training content on this website. It's multilingual, shown here in Japanese. So again, trying to reduce any barriers for users to use it by making it available in multi-languages. As emphasized on the previous slide, there's hundreds of people that contribute to this material. It cites peer-reviewed publications for primary data, and all the data and workflows are available. So they're all a subsection of the communities, but I really wanted to finish this first part of the talk with a quote from James Taylor, which is one of the Galaxy principal investigators who sadly passed away last year. But in 2019, he made note at the Galaxy Community Conference, and he was talking to the audience and passionately to the audience. And the thing he said is the most important thing about Galaxy is this community. And by this community, he was really addressing this group of people here, and this was the attendees at that Galaxy conference. And through extension, he was not only addressing the local Galaxy community that was lucky enough to come to the conference, but really all the global practitioners of Galaxy. And it is this community that really supports and makes Galaxy what it is. For which each year we try to get together and celebrate that community, learn from each other, present the latest tips and tricks and features of Galaxy. And there's been a long running history of Galaxy conferences. 2019 was the last time we were lucky enough to meet in person in Freiburg in Germany. And for the last 2 years, we have been online, as everyone can imagine. However, with no small amount of pleasure, the Galaxy community is happy to announce that GCC 2022 at this point will be live and held in America in Minneapolis. So please do chase that up if you're interested in attending one of the conferences. So I've talked a lot about Galaxy as a community, and I recognize that for potentially some people on the call, you may not have ever seen what Galaxy is, but you're going to hear a lot about it. I thought a screen grab of the service, and what better service to get a screenshot from than the relatively new Indian Galaxy server. So India use galaxy.eu, that was said in the opening statements. But just to make sure everyone's on the same page, Galaxy at this point has a fairly typical format. On the left-hand side is your discoverable list of tools, hundreds or thousands, depending on your search, or interactive data window, sorry, your history, where you keep your raw data and process data. And the middle window is interactive. The information is shown here. It's a tool setup page or it's the tool results. So that's Galaxy. It's a graphical user interface to a whole heap of compute. But let's keep talking about what it is and how we should be impressed by it. So how do we access it? And I think this is well worth talking about because my focus is on the top line. This is public servers on the web. They're open, they're discoverable, and in many cases require only a minimal amount of registration for you to get on and discover the tools or resources that that service has. So how soon can you get on it? Well, it is right now. And our Galaxy project tracks over 120 public Galaxy sites. You can install Galaxy on your own laptop, run it in a virtual machine. You can run it on the cloud, both in academic clouds and increasingly in commercial cloud instances. And I think this last one is a very important one to consider, which is you can run it on your organizational infrastructure. You can do that as quick as your own IT department works, so that could be as long as a piece of string. But the point here is that Galaxy at its core, and this is something to recognize, is an open source platform. If you want use of Galaxy, but you have concerns about data sharing or intellectual property rights around bespoke tools you've developed, then one of the solutions we know happens frequently around the world is to deploy Galaxy behind your firewall, somewhere that your researchers can access it, but not the public. But the majority of activity and the activity that's easy to track and easy to demonstrate are our public Galaxy servers. If you're interested in finding out about more of them and what they offer, then please do go to galaxyproject.org/use and you'll find a full list of all the Galaxy servers and the various places that we know, and that means that the global Galaxy knows where they're installed. So these can be very helpful, and some of them, like the Rice server for the Rice genome on Galaxy is a great one if you're interested in that particular species and want to find tools and references to support your work in that space. So we've talked about the communities, a little bit about the platform. Now, why and who do we know that comes to use it? So over the next few slides, I want to talk about who is the audience for Galaxy. We have our bench scientists who need to do data analysis. We have scientists that are doing large bulk experiments that need rigor and accessibility. And we do have bioinformaticians and computationally savvy people using Galaxy too. So for our bench scientists, well, they need to analyze their data, but they don't always have access to a bioinformatician in their lab or in their department, or and I think both the inclination and time are important factors here. Turn command line and administration of virtual machines to make them use most of Galaxy at the command line, or make the most of any bioinformatics script at the command line. So Galaxy is a great way to focus basically on doing the analysis, but without having to focus on the details of the underlying computational plumbing. We have those users that want to do large amounts of experiments with hundreds of samples and many replicates. Galaxy, through its workflows and through its quite fastidious metadata keeping, is an excellent place to promote and process data in a consistent way through the deployment of workflows. And later today, I'll be showing workflow development. And also, basically, it's also a great place to share data, because through your histories and through publishing data inside Galaxy, you can easily share the raw data, this data, and your workflows at any time. And then we have, and I think it's important to consider, our bioinformaticians and computationally savvy people. So the question is always, is this audience a good match for Galaxy? We as operators of Galaxy often find bioinformaticians saying, oh, I don't need Galaxy, I'm comfortable at the command line. And one of our counter arguments is, sure, that's great. And there are other platforms and other workflow languages out there. But, you know, we're biased, we love Galaxy, and it really is a good place to provide, as it says here, discipline to your analysis. Galaxy records your tool versions, it records your name changes, it records your file entries, exits, workflows, things that, and I'll be a little unfair here, are sometimes easy to overlook at the command line. So Galaxy does provide that rigor to your research, and I think that's a really good reminder that everyone can use the service. But if you are computationally savvy, it is also worth noting that through API access, you can programmatically access Galaxy with Blend, which means you can actually turn Galaxy into a workflow engine for your interactions, but at command line. And we do see people doing that, and we see groups invoking Galaxy from other websites as well. How does Galaxy manage a lot of this? We've heard about the use galaxy.star servers. I think they're an important thing to consider. So a number of years ago, a commitment was put in place to share resources, and Galaxy is still going down the sharing resources path. But under the recognition that we develop a tool in Galaxy Australia, there's probably a good chance that someone anywhere else in the world needs that tool. If we prepare a genome available to Galaxy, there's a good chance that someone else needs that genome. So Galaxy, through the CERN virtual machine file system, shares around the world and available, importantly, to any Galaxy service, reference genomes, tools, data repositories, and workflows in a high-speed manner so that all the Galaxy servers are synchronized with these kinds of information. And effectively, in terms of when this first started with 3 Galaxy services, it basically took our workload down to 1/3 because we could share the development and distribution of data across the global services. But sharing of data is only one half of it. Really, again, to come back to that community and to come back to what Galaxy means, it's the sharing of knowledge, which is what we're trying to achieve. And that's no better shown than through the Galaxy Training Network. We've already emphasised over a number of slides the number of people that contribute to that network. There's over 200 plus tutorials in there. There's supporting infrastructure I'll show over the next slide when we've talked about the languages. If you're interested, this has been published. So community-driven data analysis training for biology is a good read and well worth chasing up. And there are members of the global Galaxy community throughout this author list. And that is also reflected in how the Galaxy network is funded. I did want to emphasize again that global Galaxy community. So each one of these pins on this map is someone that has contributed training material. And finally, just before I move on, very successfully in early this year, the Galaxy Training Network Smorgasbord, which was a global Galaxy asynchronous online training event, occurred over a week, had just shy of 1,200 registrations from 76 different countries. It was a fantastic event to learn a lot, and that is going to be repeated early next year. So how have we maintained training? And I guess we emphasize training because there is a core recognition that many researchers bioinformatics is only one element of their overall research journey. Many are going to come, do bioinformatics when they need it, and leave again. And like any language you attempt to learn, it's reinforced by reuse. And if you're not using it, you'll forget. So training is an important part of what Galaxy can do to upskill the world's scientists. Now, any given Galaxy service, however, can be busy, very busy, or extremely busy on any given day. And that can impact on your workshop that you're running by sending your workshop users through the same job queue as everyone else. So the training infrastructure as a service, or TS service, which again, the more you spend in the Galaxy space, the more you'll hear this term, is a dedicated training queue with dedicated resources to allow you to get through your training events with the best predictability for job running time, job queuing time. You get dashboards of user progress. This was fantastic before COVID, and we all had to start working from home. It's been an absolute lifesaver for the last year or two to be able to continue to deliver training, but in a fully online environment. So I did mention 200 plus tutorials. Here's a slightly older screenshot because I know transcriptomics is now up to 30 tutorials of the variety of topics that people can access through the training network. And when you're on there, and I've sort of mentioned this before, just to show it close up, you get your hands-on slides, multiple languages, all your reference data hosted publicly on Zenodo, and you can do a quick check of which Galaxy service this tutorial will run on so that you don't start on your own server and then get partway through and realize you can't complete the tutorial. So we've talked about infrastructure, community, training. Now, once you're doing all that, how do we get from there and into the service? What do you do if you hit a roadblock? So Galaxy, I think, is quite rightly proud of its support mechanisms. And for those, we have the online forum, help@galaxyproject.org. As already mentioned in the introduction, Gitter channels are very popular. So Galaxy Project has its Gitter channel. Many of the large Galaxy servers have their Gitter channel, the training network, and so on and so forth. You'll find many support channels there. We have the community hub, which is galaxyproject.org, where you can find about the latest events coming up, which are mostly conferences and workshops, newsletters, blog posts, career center, and I think that's a wonderful thing to promote, is what jobs are available in this space, but also an insight into how the project is governed, and again, how through that governance you can influence the progression of Galaxy. Of course, globally and locally in Australia, for my example, we host YouTube channels and Twitter channels. I encourage you strongly to explore the YouTube channels. There's many pre-recorded workshop training network tutorial walkthroughs that can give you a little bit more insight simply by listening to someone talk through tools and the use. So yes, both of those, the YouTube and following on Twitter, are great ways to stay in touch with your Galaxy community. Okay, so it is quite a large-ranging talk, but it is an ecosystem and we're talking about everything. So one of the things that I wanted to mention is to come right back to the start where we talked about how many Galaxy servers and how do people access them. So tracking in this blue line here from 2013 right through to 2021 is an awareness of those number of public Galaxy servers. And there's quite a few, well over 100. There are also other deployments in a variety of systems that are coming online since that code was available. So I think rather bluntly, the next few slides are asking two questions. So how does Galaxy Project, and as a member of the greater Galaxy ecosystem, how do we benefit from having more Galaxies come on? And then later, how do each new Galaxy come on benefit from all the shared experience already? So for each new Galaxy that comes online, we'd like to think that they get a quicker startup time because of the maturity of code, the number of times it's been deployed and documented. They get access through all those support channels that we just shown to really help them deploy on their particular infrastructure or install a particular tool they're interested in. I did emphasize the access to the CVMFS repository. It's for ready to install tools. And I think a really important one, a subtle that's lost on many life scientists, which is pre-computed reference data. Best practice workflows. And again, I think I'm going to emphasize this over a few slides because it's well worth considering. You get the backing of 15 years of constant operation of Galaxy and the ability to point to a library of over 10,000 publications that have cited Galaxy in research. These are researchers that have said, I use Galaxy to achieve the outcomes of this paper. And I think that's a wonderful achievement, and it shows the robustness in the platform. So, talking about newer galaxies, well, we're here to talk about one of them today, which is Galaxy India, shown here in this pre-conference workshop now. Galaxy Africa is another example of one of the newer galaxies, both hosted at usegalaxy.eu. And what they have, which I think is important to talk about, is use. Yes, you can use effectively any galaxy globally, but what do you get out of having your galaxy? Well, what you get is a dedicated community-facing landing page. So something that you can talk to your researchers and your domain-specific, be it country or community, and direct the activity to them. Gives you the opportunities for locally targeted training courses, the operation of domain-specific tools and workflows that you can deploy onto your Galaxy service and hopefully with time through to the core offerings globally. And again, to bring it back to people, you get to identify local Galaxy champions, people that can promote the service to your local community, and you can do that through operating your community chats. So that's sort of a bit about what it means to be bringing a Galaxy online. As I said, some of these questions are deliberately blunt or selfish. So what does Galaxy Project get out of all the extra galaxies that come online, apart from the feel-good factor? So what we get, and again, it's blunt, is by having members of the global Galaxy community give back. One of the core things that makes Galaxy successful is the large scope of tools that are available, back from well-embedded historical tools that are maintained through to the latest publication that someone quickly wraps and makes available for Galaxy. So yes, if you're in Galaxy, this is almost a plea as well as a request, which is look to develop tools. Take your tool of interest and promote it to the global community. Through that, not only will you get potential troubleshooting and tool improvements, but also I think it's important to recognize that you will get personal recognition for deploying tools in this space. You get access to training and shared experience, which is what Galaxy Project gets, which is code development, observations on tool usage, tricks and tips that you learn from your Galaxy that you feed back to the rest of the world. And I think also a very important one, trends and observation in usage, by which I mean what kinds of tools and data analyses are people performing that they weren't performing maybe 12 months ago, and what does that mean for your Galaxy instance, or what does it mean for Galaxy Project in terms of refining the code base or refining resourcing. And again, you can contribute. This is from the training page, but do explore either of these web links to see how you can contribute to Galaxy. So that's been a lot about infrastructure, compute at a high level, and I recognize that. I wanted to change in the last sort of part of my session to really focusing on what kind of science does Galaxy do. So there is just going to be by example, and then we'll finish with some other content. So as I've alluded to before, somewhere around, I think it was mid-2020, Galaxy Project's tracking of publications past 10,000. We're now past 11,000 and edging our way towards 12,000 publications. And you can see that growth over time. Very satisfying, and growth over cumulative and per year. It's an easy to measure metric, but for many of us in a grant cycle, you're putting down your papers to justify getting funding. Galaxy project is no different. To be able to show what science we enable is one way to continue the funding in, to continue to deploy the platforms regionally and globally. So yeah, fantastic numbers to see. What it is Galaxy has spent time, and Use Galaxy Europe is probably the champion of this at this point in time, is making domain Galaxy available to researchers, so that, again, like the community-driven Galaxy pages that are regional, we also have community pages of practice. This is the High C Explorer for looking at chromatin configuration tools in Galaxy. We have a single-cell Galaxy for exploring single-cell RNA-seq data and the quite specific tools that are in that domain. We also have, outside of our genomics domains, increasingly Galaxy's coming online for supporting other styles of life science. And Galaxy P for proteomics and metaproteomics is a great example of this. They have multiple subdomains, but all can be found at galaxyp.org for proteomics. So a lot of mass spec based analyses. So the next example, and this is one that is very much global, but as I said, I will be putting the odd Australian spin on slides, is SARS-CoV-2 analysis. So with the initial rush of sequencing of samples a few years ago, when the first outbreak globally became aware, the bedded down best practice workflows, data sets, and reanalysis of potential COVID carrying patients in a human mixed sample. The landscape was ill-defined. It's probably the nicest way to do that. And the Galaxy project really stepped up and took to this challenge and decided, if we can present a series of best practice workflows, we can deploy these globally and make it available for any researcher, clinician, pathologist lab to analyze their data and help really give a clearer global picture to where SARS-CoV-2 was at. So there's a number of papers in this space. There's the initial analysis paper here. This has been followed on, as you'll see in the next slide or so. In Australia, we were lucky to be supported by both of our supercompute centres in Australia, who spun up a largely resourced VM that Galaxy could access to basically push through 6 different styles: analysis, evolution-based, chemoinformatics, proteomics, Arctic for amplicon-based searching, and direct RNA-seq in a globally consistent workflow manner. And that can all be found at covid19.galaxyproject.org, where a number of workflows have been made available for you to execute on your data. Certain Galaxy instances, and Galaxy Europe does this, automatically ingests any new data sets uploaded to online repositories and pushes all their data through this for doing consistent and up-to-date SARS-CoV-2 mapping. And how did we do it? Well, we did it in Australia by taking all those workflows and making them available on our Galaxy service for anyone to use and reuse, just as the other Galaxy services have done. So the next example I thought I would give, and this is both a biological example and it will become a technical example. So as part of the Vertebrate Genomes Project, which is a global endeavour to provide a genome sequence to approximately all 70,000 vertebrate species known, Galaxy Australia has been engaging with VGP and international galaxies to see how we can support these efforts. So first of all, the Vertebrate Genome Project deposits all their raw data in a freely accessible GitHub location, also incidentally one that is accessible inside Galaxy, so you can download through Galaxy from here without having to move your data around. We have well-described publications, this being the most recent one from earlier this year, for describing the workflows that the BGP are advocating for doing genome assembly. And on GenomeArc, we have a long list of species with varying levels of data. So Galaxy and Galaxy Europe, and I'll talk about Galaxy Australia, have been looking to convert those pipelines as presented in publication in Galaxy workflows, enable the tools for those pipelines, and have also made a wrapped site, which I'll show in a minute, for accessing BGP. In Australia, we've been replicating these workflows and tools onto our national service. But as I've talked about community-specific or domain-specific environments, we actually have an Australian community under our Threatened Species Initiative, for example, who have a specific interest in genome assembly. So we've been taking the BGP pipelines and configuring them for our local communities. And I'll show what we're being able to enable with some large resource allocations. So here is the European website, assembly@usegalaxy.eu. And again, you're familiar with the content now. Tools, history, and this sort of domain-specific landing page in the middle. So that's what Europe's done. I thought I would show a real tangible example of what we've done in Australia. We've got some news articles on this. But as I talked earlier about how do all the galaxies benefit from each other? Well, one of the trends that we saw in Australia was the increasing use of Galaxy for data-intensive jobs. Larger data sets, requirement for more CPU, and the requirement for more RAM. So we took this to our funding bodies earlier this year, and they were very generous in enabling the purchase of 5 high memory, high versatility nodes. So these have a few hundred very fast CPUs, 4 terabyte of RAM, and some real stunning local storage with high input-output capacity. And we've been turning the attention of these high nodes to doing genome assemblies. So within Australia, one of our bodies, Bioplatforms Australia, has a repository that hosts framework, so data that is pivotal to Australian management of Australian species, and that includes the genomes of Australian plants, AusArcs, the Australian Amphibian and Reptile Genomes Project, and the Threatened Species Initiative, to name a few. So we've taken data, in this case the AusArc data, through a pipeline much like the BGP one that does some data conversion, some assembly, and then a whole slew of post-genome assembly quality control. And we've made this available to these communities of practice here, many of which fit into that very first scenario I presented much earlier in the talk about biologists that are not bioinformaticians. So these are people, in many cases, not even in academia. They're in zoos, they're in environmental management, they're in government, but they need genomic data to help them make decisions. So by enabling a workflow like this in Galaxy, with Galaxy's relatively simple play button for a workflow, they can get quite a complex analysis done for them. How did this impact us? Well, we generated genomic sequence from one particular threatened amphibian species. The right-hand side shows a bandage image, which is a representation of the contigs assembled. They're very clean with very little misconfiguration, which was great to see. Have a reasonable amount of contigs, a great total length, and a high N50. But what was also, to me, very, very satisfying was we were able to make use of our high-end infrastructure to do this assembly in 2 hours from start to finish, which in itself is great, but also for the iterative process that's genome assembly, this is fantastic. It means a user is not spending days or weeks waiting for a result before they have to reiterate it and try to get the process back into their memory. In 2 hours, they can have another go, think about their settings, what it means for their genome, and get rapid turnaround in really important biological questions. So that's our user examples. I have a few minutes left, I believe, and I want to wrap up before we go to questions. And this is actually a very true slide, late breaking news. So the September release of Galaxy has only been out about a week or so. And one of the elements of my job, one of the more exciting ones, is to get to go through the code release and find out what that global Galaxy community, listening to all the practitioners from around the world, has deployed in the latest version of Galaxy, and then what does that mean for us to be able to give back to researchers. So my team went through this for me and have extracted out some of the really salient but new features of Galaxy that I think are worth talking up. And in this case, please do find all of the latest features at this much longer web link down the bottom here, but you can just Google Galaxy Project 2109 to find this link. So some new features in Galaxy to talk about, ones that I think are very important. And the top one I've put here, which is our, in quotes, new user welcome page. And for this conference in particular, where we're talking about the soft and then launch of india.usegalaxy.eu, this welcome page I think is a fantastic addition to the software code. So what it is, and I've shown only a part of the new landing page here, is the first time a user logs into Galaxy, rather than being presented with that middle page, which is very information rich, but often very information rich for a seasoned practitioner of Galaxy. The first time you log in now can trigger a totally new landing page with some really basic 101, how to get to know Galaxy, how to move around the service. This landing page is configurable. So again, you can put in domain-specific knowledge about how you want people to move around the service. So I'm ridiculously excited about this new feature and can't wait to see it in the next few weeks on Galaxy Australia. We have new tool panel filters, including EDAM categorization. I'm going to come back to this one because I have a whole slide dedicated to this. This one I think is super exciting too. So one of the things that Galaxy Australia did, and I know the other Use Galaxy servers have enabled it to various extents, is a recognition that Galaxy is an analytical platform and not a storage platform. So we want users to move their data in, but also importantly to move it out when they've finished their analysis. And in many cases, depending on your grant funding or your institutional requirements, you are actually required to keep your analyzed data for a number of years. So moving data in and out of Galaxy, to be blunt, 15 years ago wasn't so much of a challenge because data set sizes were smaller. 5 years ago, not so much of a challenge. Certainly in the last few years with the advent of long-read sequencing, moving data in and out of Galaxy, we understand has become a bottleneck for people. So 2105, the earlier release this year, enabled the much more seamless import of data from cloud resources, which was fantastic. But now you can export back to cloud resources. And I think this is just great. We enabled it in Australia last year, and I think helped prove the proof of concept for Galaxy Project to take this work on. But it's a really fantastic feature. Tools always grow, but with tools requires data types, so it's always just great to see that Galaxy is supporting more and more data types. And for those of you sort of interested in Galaxy under the hood, I think two very important improvements and a resilience improvement in the uptime of the web service and pulsar compatibility. And we haven't chatted about pulsars today, but these are remote worker nodes of Galaxy that allow Galaxy to handle more jobs than the single substantiation that is the landing page. They are complex, but well worth having. So this as a statement is a great one. So on my next slide, and this will be my essentially second to last slide, I want to talk about this tool panel filtering. I'm going to do this through a video that Galaxy Project has put together showing this feature. But just as a context for this, one of the pieces of feedback we know we get on Galaxy Australia, we have 1,500 tools installed. We could install 8,000 tools. Is a user find a tool? And in many cases, the definition of the tool, is it a QC tool? Is it an RNA-seq tool? Is it an ecology tool? Is up to the author of that tool, which does produce some human variability. So I think having something like the EDAM ontology, which is a structured ontology associated with biology on the tool list, is a fantastic way to allow users now to more intuitively explore Galaxy, find tools what they want to do, and not be left guessing if they're looking for the right tool. So I will show this video. It doesn't have sound. I'll probably talk over it, but let's see if we can get this to work. So here on the left, we're looking through the typical tool menu and we're flipping to ontology, and we're now looking at much more clearer statements, sequence editing, sequence cutting, sequence trimming, hopefully more human intuitive tool descriptions rather than the original language presented in Galaxy. And these ones, even better, now by domain. Microbiology, gene analysis, bioinformatics, which I guess you would hope is on there. So this is a new feature. We'll see how it plays out in Galaxy, because for ourselves, we'll be installing 2109 in the next couple of weeks. But I think it's a really great feature, and it speaks to the core of how Galaxy listens to its users to try and improve the platform, so that if, as a new user to Galaxy, you shouldn't have to go away and read 15 tutorials about how to get the most out of Galaxy. Hopefully, it is almost self-explanatory for you in the first instance. So with that, I'll wrap up. This is a very biased slide from myself, but I did give you that heads up. So we now view Galaxy Australia as a critical digital research infrastructure for the Australian life science communities, and more broadly as part of this global Galaxy ecosystem. And so I think that's no better exemplified than two recent Nature publications in Nature Biotech and Nature Method, both in the latter half of this year, that cited Galaxy either in the ongoing monitoring of SARS-CoV-2 with specific mentions of Galaxy Australia, or in this Nature Methods paper that compared bioinformatics workflow managers and very nicely and clearly put Galaxy alongside other workflow managers. But either case, you know, we're getting talked about and published in Nature, and by we, I mean Global Galaxy. And I think that just shows the maturity of the platform. So with that, we're at conference mode, and so Galaxy Australia is definitely keen on hosting and planning to host Galaxy conferences. So I do hope that the next time we do this might be when I'm presenting to you live in Australia. So in the meantime, however, please do go and put Galaxy to use at india.usegalaxy.eu. And with that, I will thank my quite large team of Galaxy developers, trainers, codes, and everything else in between that's Galaxy Australia, and all our funding and support partners at the bottom there. Thank you very much, and I'm more than happy to take questions.
Thank you, Gareth, for the nice presentation of introduction of everything. That's definitely needed for this workshop, because many people are pretty much new, and so this will help them. So this is now questions time. I'm checking both the places if they have any questions. So it's a nice time to ask questions if you have any Galaxy related questions. But meanwhile, until the questions comes up, so I'll go with my questions. One of the question could be like when people comes to the Galaxy that there are multiple instances of Galaxies. So which one do I use? Like, why should I go to which one? So that's kind of a confusing question. Some people, yeah.
That's a great question. And you're absolutely right. So I would say, and I'll try to do this quickly, a number of years ago, that was a very difficult question to answer because no Galaxy service really looked like any other Galaxy service. If you discover by Googling or by reference the usegalaxy.star servers, you can be relatively ensured that they have the same toolset, core toolset, the same reference genomes, and are supported behind the hood appropriately, as in resource sufficiently to run any data. So that's one answer. If you're at a usegalaxy.star service, you can pretty much use what you want. However, it is worth also recognizing on the flip side that many of the Galaxy servers that are public are there funded to provide to their local users. So Galaxy Australia is primarily funded to enable Australian research, but we're open to global collaboration. Galaxy Main, Galaxy Europe, all the same. So I would say flippantly, whichever Galaxy you land at is the one to work at. However, if there is one associated with your region, your community, that is generally the better one to go to for help desk engagement, quota discussions, and tools.
Yes. So does this guarantee anything like all the use Galaxy Star has some kind of minimum number of tools that would be there? Is it something like that?
Yeah, so yes, there is a core tool set that comes out with the Galaxy Core, which is, I think, about 800 to 1,000 tools. There's a commitment by the usegalaxy.star servers to support the Galaxy training network, and I think that's a really important point to emphasize, is if you want to do training, be on one of those servers, all the tools and workflows will be supported. Beyond that, yes, you're then going to get into your domain-specific servers that really generally have less tools. Doesn't mean less functionality, just tools that are specific for that domain. So if you arrive at the high C domain or the RICE Galaxy, you're going to get tools specific to that community. By the flip side, I guess I would say the person that we're always most interested to hear from is the researcher that says, I can do 90% of my work, but wouldn't it be great if this tool existed on Galaxy, because then I could have an end-to-end solution. So we're always on the lookout for what tools to install on Galaxy. It's the best way we get feedback about the scope of the service.
Okay, great. I'm also enabling the chat in the Google Meet. Probably if you're facing difficulties in other platforms, you can also write in the Google Meet chat as well. And also you can raise your hand so that I know that you want to ask questions. That's also a possibility. Okay. Meanwhile, any question comes up, I will go with my next question. Okay, probably there is some question. Okay, then I take that one.
There is one. I'm happy to read it out. It says, hello, thank you. Suppose I want to make changes to my Galaxy portal for a particular job pipeline. Is that possible? What if I wish to add my own codes to Galaxy? Is that allowed? So yeah, look, this is a really great question, and thank you for it. So there's two parts here. I'm not entirely sure about making changes for a particular job pipeline. So what I'll do very quickly is describe how Galaxy works. So a combination of mostly three things: your user ID, the tool you want, and the size of input data dictates where that job will be sent on the infrastructure. So within Australia, we have computers scattered across our country, and that combination of mostly tool and data set size says, do we send it to a 2 CPU machine, a 6, 8 CPU machine? Do we send it to our high memory node with all 256 cores? So that particular aspect of Galaxy is generally run by the administrators of the service. Two things will happen. A user will bring a lot more data than we recognize, and the tool will fail over. It will communicate with the user that it's too much data for that tool. And that generally, even though it's going to sound like a reverse logic, is often the best place for a user to engage with the service and say, I really need to run this tool on this amount of data. Can you do something? And the short answer is yes, we can. For you as an individual user, for you as a group of users from a particular university or group, or for everyone, we can change that combination of rules around resourcing and re-resource it till it works. And that's why we bought in Australia our high memory nodes, because we were reaching that upper capacity to resource particular tools. Can I add my own codes to Galaxy? Is that allowed? Well, that's actually a really good question again. There's a long answer. So depending, I would answer it multiple ways. One, talk to a practitioner of Galaxy. It may be that your code is doing something that we also know how to translate into pre-existing Galaxy tools. That's one. Two, if it's code that's applicable to other researchers, then we would very much encourage you to wrap it into a Galaxy tool, and then it can be installed on multiple Galaxy servers for you and others. And the third one, which I haven't chosen to emphasize today, it's a relatively new feature of Galaxy. It's called an interactive tool. So it is possible inside Galaxy to spin up, for example, an RStudio and do specific data manipulation inside that RStudio, bring your data back into Galaxy. So that is one of the avenues that bespoke code can be provided into Galaxy. And I hope with that I've answered the question. I can see now, thank you, that the chat's enabled. We have a second question. What if I want more space on the Galaxy web server? What is the procedure for that? Good question. Depending on your Galaxy server, and I won't attempt to share my screen, on Galaxy Australia, at the very top of our screen, we have a button that says Request Data Quota Increase. The same will be of the other Galaxy servers. Simply contact the service and say, I would like to bring in 500 gig of raw data. Can I please get a quota increase? And yeah, that will almost always happen is the short answer. Pardon me. Question from the audience, is there any module for variant interpretation? That's a good one. I would say on the Galaxy Training Network, there are a number of tutorials for variant description, as in how to determine variants in haploid or diploid systems in complex cancer models. In terms of variant interpretation, there are some tools that will allow you to interpret the consequence of that variant in terms of protein change. If what you're looking for is more a CAD score or some kind of metric of pathogenicity of the variant, no, I don't believe there are any on Galaxy, but there are other online resources you can use.
Okay, that was the question. So go on with your more questions. You can also ask questions related to the registering to the Gitter and other things, but of course, right now the priority would be this session questions. Okay, I go with another question. Sure. So you said in the new update there is a facility that you can the tools can be recognized. Can you more elaborate on that? Like, is it expanding to all the instances, or is it only for the Use Galaxy EU is happening?
This is the EDIM ontology tool description. Is that the bit you were?
Yes.
Yes. So no, that's going to come out with the latest code base. So that, well, the political answer I'll give you is that will be on any Galaxy server that people maintain up to the latest version. So again, all the large public Galaxy servers have an informal agreement to install the latest version of Galaxy once it is released. That way, again, a user that is on different Galaxies or talking to a collaborator about their experience on one Galaxy to another have a uniform code experience. So the very short answer is yes, that EDIM ontology functionality, it's not on all tools yet. It's on a growing list of tools, but it will be on all Galaxies reasonably. And by reasonably, I would say For example, in Australia, we put it through a development, staging and production process. So that takes a couple of weeks. So we're anticipating it'll be on Galaxy Australia by early December.
Great. So I'm just looking for other things if there are any questions.
I believe one just popped in. Is it possible to develop Galaxy as an operating system with all the mentioned features for local machines? I think the answer is yes. So it is possible to deploy Galaxy locally as a virtual machine on a Windows or Mac computer, or deploy it on a Linux machine. Certainly the way my team of developers add new content to Galaxy is they have a local substantiation of Galaxy on their machine, and they're interacting that through local hosts. So I believe the answer to the question is yes, and it will have all the features that Galaxy has to allow you to run it locally. It also, given the speaker and their initial question, yes, it gives you the potential to add any of your own code into Galaxy to see what it does as tool functionality or changes to the code base.
Great. So I have another question. I think the European Galaxy and then Australian Galaxy are more using as in the pulsar thing. They exchange the jobs. That's what I understand. So how was this collaboration actually? So I mean, so let's say if you want to talk about any of the difficulties or maybe some things, what could kind of happen?
Sure. So yes, Galaxy Australia and Galaxy Europe, well, all the major galaxies can make use of pulsars, thanks to the way they're funded and distributed in the country. Certain countries have made more use of them. I think I do need to talk relatively bluntly about what happens under the hood with Galaxy, and I think it's something that is good to make people aware of depending on your domain. So your data moves to wherever the web page is hosted. Then it moves from the web page hosting site to the Pulsar site. It's computed and the results are sent back to where the web page is hosted. So for example, in Australia, all of our Pulsar machines are hosted within national borders. And this, for a small percentage of our users, is very attractive because of the way that they are funded. Their data sovereignty is something that they need to maintain, and national borders are a convenient data sovereignty boundary. For other pieces of data, that's less of a concern, and you can enable a Pulsar to be anywhere in the world, actually. And we have made use of Galaxy Europe. We send data to Galaxy Europe sends data to Australia, and we do that to enable large training events globally. Either a large training event nationally where we don't know if our national infrastructure we support, we overflow to Europe, or vice versa. So the Pulsar network allows basically an overflow system. So once your primary system is full, you can overflow to Pulsar. You can also add into your Pulsar system machines that you wouldn't normally have sitting close to your landing page. So our Galaxy operation, which is hosted at one of our supercompute centers at Pawsey, has, and this is not rude, it has very generic flavors of machines, very normal ratios of CPU to RAM, which works for most bioinformatics tools. But for those tools that are very CPU intensive or very RAM intensive, having the Pulsar network is a fantastic way to enable those jobs to run to completion by sending that job to a remote system that is built for that style of work. So yes, that is something that Galaxy Australia, Galaxy Europe have done quite well and collaboratively done with each other. There is actually a growing vision for Galaxy Global to have a shared Pulsar network globally. And again, I think that's a fantastic goal to have. I'm quite confident it will happen. I think there will be just a couple of nuances around data sharing to sort out first.
Great. So there is another question. You can take it from the chat.
Thank you. I think I just saw that come in. As Galaxy Australia is working on SARS-CoV-2, is the data publicly available on the Galaxy platform? No, that's a great question, and unfortunately I'm going to say in this case, no. We have enabled the workflows and the environment for our users to use it, and we do know our public health laboratories are using it. It is unfortunately then up to their decision as a user to where to place the data. So I would highly recommend, if you would like that data publicly available, go to the COVID-19 Galaxy project resource, or go to Galaxy Europe, who I know are doing a lot of the heavy lifting of turning that into the public resource. It will contain Australian data, but from our particular point of view now, we're not making a requirement on our users to publish their findings through that workflow.
Okay. Thanks, Esther. So more questions we will be waiting.
I'm happy to take more.
Just
Kiran,
please go on.
can I ask a question to
Yeah,
Professor?
please go on. Yeah.
Sir, there are several young software developers across India, and can you please advise them how best is to develop software which is Galaxy specific, and then which enables them to publish papers also? Yeah.
Absolutely. Really good question. So I guess I have encouraged how to turn a tool, have tools in Galaxy, but haven't mapped it out. Look, I'm going to, when I get a chance, I'll find it on my second screen and paste it in here shortly. The very easy answer, go to the Galaxy training project site, so training@galaxyproject.org. There are whole tutorials on how to make tools available to Galaxy. Generally, most of them are based on a core GitHub location, which gives that person a digital object identifier or a DOI. Publication route from there is multiple, so obviously they can publish themselves. The tool can get into the tool shed, it can be publicly cited, and from there, certainly as a service, we track tool usage, but there's actually no reason why a user couldn't write to a service and say, could you please tell me how frequently this tool is used? So I hope that helps, but the core answer is training@galaxyproject.org. Has tutorials on how to rack tools, and that will be the best primary place to go for that information.
Thank you, sir.
Not a problem.
There's another question, Gareth, from what are the modifications according to you that can be implemented in current version of Galaxy?
I'm not entirely sure what's meant by modifications, sorry. So improvements, potentially, I could address, but yeah, I don't know about modifications, sorry. Maybe if the user has time to just type out a clarification, and I'll keep an eye on it.
Lipsia, you can type more clarification on the question.
That's right. I'm actually just looking up the how to write a tool bit on Galaxy as well, so it gives me a second to do that. I mean, look, at a very high level, Galaxy is always under improvement, and it needs to be, because the community we're servicing is changing. It used to be predominantly short read sequencing and very short read sequencing. Then it is a combination of short and long read sequencing. And now it's not even just genomics. It's now proteomics and metabolomics. And thank you, we've got what are the advanced modules that can be implemented further? Thank you, I think I understand a little bit better now. So one of the things that Galaxy is aware of and this is where this sort of trending for the community comes in, is there seems to be an intense interest in machine learning, deep learning, and artificial intelligence at this point in time. What that is meaning behind the scenes is access to GPU units, so graphical processing units. They are putting interesting demands on how we run Galaxy. In Australia, we are doing this through an exploration with a number of the commercial cloud services, and commercial cloud is also going to give us an opportunity to allow potentially institutes and universities to bring compute to Galaxy. So the future modifications around Galaxy are going to be about bring your own compute. So I have some spare compute and I want to link it in. Bring your own data. I have data somewhere and I would like to bring the compute to it. And through Galaxy Global and through a process called Anvil, Galaxy Project is exploring how to substantiate galaxies in a transient manner. So they're created and destroyed as needed for handling of sensitive data, in particular human genomic data, so that data sovereignty is a real protected environment. So there are large changes, I believe, coming in the next few years to recognize all the different ways that people want to utilize the platform. The link also I just dropped in before is to the developer section of Galaxy, and it has 5 tutorials on tool development. We have another question on SAM tools, MPileUp option. The storage exceeds for large files like human data. Is it possible to increase the storage space? Thank you. Again, yes, you will need to speak to the administrator of your specific Galaxy service. And if we have a few new people here today, as I understand we do, I'm just very quickly juggling between my 100 open screens. Apologies for this. I'm actually trying to look for a Galaxy error on my page. This may be just best shown by example. I'll just find our presentation. Apologies for this. Yeah, I realize I'm lagging a bit because I'm jumping between multiple screens.
That's fine, yeah.
So here is India Use Galaxy. Here are some tools that haven't worked. And again, this might be coming up. If you ever run into an issue in Galaxy, you'll get a red box. They're somewhat depressing to see, but that doesn't always mean too bad a thing. Under here, you'll have view or report this error. It will tell you the error. This one's not particularly verbose, but you can report it. You get an opportunity to put something in. I tried to analyze 4 terabytes of data. Please help. I'm not going to hit submit because this will get to Galaxy Europe. But this would be, to me, the most prudent way of troubleshooting a tool or an observation you have, like the mtools, SAM tools pileup question, is simply if the tool errors, report it to the Galaxy instance and work with them to see if they can change your configuration.
So I'm just looking if there are any more questions. Okay, please go on, Harpreet. I think you are host. You should be able to ask. Let me see if you're not. Yes. Harpreet, make yourself unmute or maybe type the question. Oh, okay.
Thank you, Kiran.
Harpreet, you're in multiple devices. Maybe you can.
Yeah, just give me a minute. My voice is clear now.
Yes, Harpreet, go on.
Thank you, Gareth, for the wonderful talk and
My pleasure.
taking up this challenging task in for Dave. And I have a question. I just have a different question to you. How do you see the role of regional galaxy communities in future? We are trying to develop very regional galaxy communities, like Galaxy India. We are trying to build. So, what do you see the role of regional galaxy communities in the future? How they can be benefited, and how they can be, you know, uh, how they can contribute also to making the galaxy even more useful.
Um, great question, Harpreet, and thank you. Look, the short answer is I have seen before Galaxy, well, before I knew about Galaxy, and certainly since then, within Australia, other virtual laboratory environments try to start, generally in different domains like ecology, imaging, etc. And one of the things that they come to Galaxy about is, to be blunt, how are you so successful as a global service? And the reality is, if you go 15 years or potentially even 10 years, there wasn't a certainty that Galaxy was going to be the successful service. We can now look back and be very proud of where we got to, but at the time we weren't certain. And really, to me, and again, I sound like a one-track story here, it is about listening to the community. So with each new Galaxy that comes online, it's an opportunity for researchers to directly engage with bioinformaticians in the way that you would never write to Microsoft and ask for a new feature in Excel. You wouldn't necessarily write to Qiagen and say, I love CLC genomics, but could you put this new tool on? Because the interface is just too far away. With each of these galaxies that come online, we get a new insight into how that domain needs to operate, the kinds of data that they're working with, the kinds of genomes or proteomes or image data, and each of those just simply informs the product better. So it is very high level, but I think that is how each of these galaxies make it best. I hope that's answered the question.
Thank you very much, Gareth. Thank you. Thanks for the support you have been providing us from the beginning. Thank you very much.
Our pleasure. We do have one more question.
Another question.
Yes, I
That's
just saw. Thank you. That's come in. Is it possible to host data sets on Galaxy? And really, each time one of these questions comes in, I realise there's something else I didn't get to say. So apologies for that. The short answer is yes, absolutely. Galaxy would love to host data. We host reference genomes because the way a tool interacts with a genome, it often has to do a lot of pre-work on that genome file to make it available. We've done all that pre-computational time for you, so when you see a genome available, it's immediately available. But databases are outside just the genome. So I more broadly should talk about reference data sets, not so much reference genomes. And yes, there's no point in filling your particular history with your reference data when you should encourage your local Galaxy to host it for you. The management of your quota is better, and the basis as well is that almost certainly someone else will benefit from being able to access that data too. So yes, please don't suggest you're going to put up your database of your own PhD information, your own intellectual property information, because if it's in the shared repository, everyone can access it. But if you identify an online repository which you think would be useful for others to access, please, again, contact your local Galaxy service, ask them to host it, because that is the most efficient way to disseminate that data.
Hey, great. Okay, great. So I think we are or more than the time. So that shows the enthusiasm about the galaxy. That's very good. Thank you very much, Gareth, for the session and also for answering all the questions. And I thank all the participants also for actively taking part in asking questions. So this session ends, but of course, we will stay online as of now for the next session. And then the next session starts in another 10 minutes. You can still ask other questions related to the gitter, although I think some people are not still familiar with that. So please do ask about that. And we will still try to answer those things until the next session starts. And we will stay here right now. Thank you, Gareth. And
Thank you.
yes, Jeremy, sir.
It will be a great thing to ask everyone to unmute the video and then take quick snapshots?
Well, I don't know, 60. I'm not sure if it's possible. Okay. Let's try.
Renuka?
Yeah. Yes. I think we should try. Thank you. Gareth, can you please also stay back? Yeah.
Absolutely.
Yeah. So I'm enabling the video. So now the video is enabled. So everybody can enable their videos. So I'll still wait for more videos to come up. Well, it's coming up. It's very good. It looks like it could fit in one screen, but let's see. So only 35 to 40% got open. I'll wait for another minute. Please open your webcams so that we can see your wonderful faces. Okay, now I take the picture. Okay, still they're coming up. Okay, fine. Good. So in a count of 3, I will take the pictures. So you can show your smiley faces, even though if you don't like me. 1, 2, 3. Thank you all for the wonderful picture. So we have a picture. Yeah, thanks. So as said, you can still keep asking questions about anything related to the Galaxy. If you're making an account, if it's the first time that you're making an account, also about the Gitter thing, you want to ask questions in there. And yeah, anything you can ask that. Thanks, Gareth, and we see you in the next talk again. Yeah.
Thank you. Yep. See you soon.